Let us now look into a example for repressible operon. The example that is commonly given for this is the tripped operon which is involved in the synthesis of an amino acid called tryptophan. Again, in the bacteria E. coli, synthesis of tryptophan requires 5 enzymes designated as A, B, C, D and E. Therefore, there should be 5 structural genes involved in this mechanism, one each for A, B, C, D and E. So this is the DNA molecule. Let us say in this DNA molecule there are 5 structural genes. I will designate them as S1, S2, S3, S4 and S5. This is the operator gene. This is the promoter gene, to which RNA polymerase molecules are attached. This is the regulator gene. So as in the previous case, regulator gene synthesizes mRNA by transcription. This mRNA by translation synthesizes a protein, and this protein, let us say, becomes a repressor. Let us start with the assumption that there is no tryptophan available in the cell. Tryptophan itself is the co-repressor. So co-repressor is not available. In the absence of the co-repressor, the repressor becomes inactive. So when this repressor is inactive, it cannot bind with the operator gene. Therefore, RNA polymerase molecules can reach the structural genes. So each of these structural genes now enter into transcription. 5 mRNA molecules are synthesized. Each of these mRNA molecules in the ribosomes undergo translation. So 5 protein molecules are synthesized. 5 protein molecules are synthesized. And these protein molecules undergo the necessary changes to become the 5 enzyme molecules A, B, C, D and E. So all the 5 enzyme molecules are now available in the cell. Therefore, the cell now actively synthesizes tryptophan. The genes have been switched on. So tryptophan is now available to the cell. So the cell starts using this tryptophan to build up its proteins. Suppose all the tryptophan molecules are utilized. So sufficient tryptophan molecules are synthesized now. Now one of these tryptophan molecules binds with the repressor, begins to function as co-repressor, makes this repressor become active. So repressor binds with the operator gene and the mechanism gets switched off. So let us see what happens. DNA the 5 structural genes operator gene, promoter gene, regulator gene, has synthesized repressor. So repressor is there, but co-repressor is absent. Now tryptophan is synthesized. So you have tryptophan molecules tryptophan is synthesized. One of these tryptophan molecules binds with the repressor, binds with the repressor. This is repressor, this is co-repressor. So now repressor becomes active. This repressor can bind with the operator gene, thereby prevent the flow of RNA polymerase. Therefore, no transcription, no translation, no proteins, and consequently no enzymes. So the synthesis of tryptophan stops till all the tryptophan molecules are used up again. Co-repressor is absent, again the structural genes are switched on. So this is when co-repressor is present. So this is the mechanism in repressible operon.